phosphatase. You have to have a phosphatase to take it off. And so GTPases are very different that way because they're much more of a timer as well as a switch. So they turn themselves off. Both, of course, have enzymatic activity. So I will now select the answer on here that you can't see, but it will count. If I can get this whole thing to work here. And I'm sorry about the technical issues. It seems that by the end of the term, I'm sure we'll have worked all of them out. So our next Later question is a ligand that binds to an enzyme other than in its active site and stimulates the enzyme activity is called a allosteric inhibitor, allosteric activator, competitive inhibitor, competitive activator, or substrate.
Yeah. What does that mean?
Oh, I turned off polling. Oh. Sorry about that. We'll start this again.
It's like a soccer.
Somehow again, technical issues are happening again. Please vote again on this question. You've voted once already.
Definitely needs some coffee after this. I think for the test for lab today, we need to learn a different term names for the bio quiz. I think we will read it. I think she's gonna just.
Do we really need more time? Yes? Okay, stop now. I'll look here at the results. It looks as if 87% of you agree with me. It's an allosteric activator. Again, either your previous instructors did a good job of telling you about this, or you listened last time. So as our third clicker question, we have the covalent modification made to proteins that get degraded in the proteasome is binding to GTP, hydrolysis of ATP, addition of a phosphate, addition of a polypeptide, addition of a polysaccharide. Share between yourself.
Did you just I have no idea because I don't remember. I'm going to say phosphorylation. Just because what is the answer? I have no idea. It's addition of a phosphate. I wrote something about it, but it doesn't. Is it B? C? D? I don't know, I picked C. You pick C?
Okay, let's stop. I'm guessing, based on your results, we've got 9% that like A, 27B, 27C, 32D, 5% E, that I didn't do a good job of explaining this. Because we seem a lot less clear about it. So let's go through these individual possibilities here. Covalent modification against the proteins to get degraded in the proteasome is what? Binding to and making a covalent addition to ubiquitin. What's ubiquitin? It's a protein, also known as a polypeptide. So it's D. There is binding to GTP, of course what's happening with G proteins. Has practically nothing to do with the proteasome. Hydrolysis of ATP, on the other hand, does have to do with the proteasome. And that's because you need ATP hydrolysis to break down all the proteins once it gets inside the proteasome. And you also have ATP hydrolysis in getting that protein into the garbage disposal part of the proteasome. Addition of a phosphate, again, these are kinases, has practically nothing to do with the proteasome. Talk about addition of polypeptide, that's ubiquitin, and polysaccharide modifications. We're not going to talk about in this class. They're frightfully important and interesting, but again, beyond the scope of what we're talking about. So we talked about last time, again, we had some other folding enzymes. These were your peptidopyrolysis transisomerases, the disulfide recombinases, etc. Looked a little bit at ligands, particularly in terms of the enzymes. And then in terms of regulation, this is really the regulation of enzymes, but also talking about the regulation of protein stability, particularly in terms of the proteasome and degradation. We have questions about that before we move on and continue with today's lecture? No. Okay, so we'll finish up talking about the proteasome today, and then move on to some of my favorite, or actually maybe now least favorite kinds of protein methods, because I just found out that one of the methods I was trying to do this morning didn't work. So a little bit about basically how you go about purifying proteins. You think about a cell, it's a mixture of thousands of 10,000 different proteins. Usually to understand what one is doing, particularly the enzymes we talked about last time, you need to have that particular protein separated from all the other ones. So you can understand what its KD is, what kinds of catalysis, what kinds of substrates, what kind of products, etc. So that process of protein purification is really critical in terms of understanding how those proteins are actually functioning. Talk a little bit about how we do that. Very broad brush kinds of aspects, chromatography, electrophoresis, and then in terms of analyzing those individual proteins, particularly analyzing a whole bunch of proteins at the same time, there have been amazing developments in mass spectrometry in the last couple of years, and it seems like every couple of months there's a new mass spectrometry technique which has come along in terms of trying to understand what's happening in terms of not so much individual proteins, but very much all of the proteins in a particular cell, particular organ, subcellular component, etc. So the developments in mass spectrometry have really kind of revolutionized what we know about proteins, and particularly mixtures of proteins, which is really what we have in cells. And so that process is also known as proteomics. So let's finish up talking about the proteasome first. Again, it's the molecular garbage disposal right here in the middle. This is the area where you have ATP hydrolysis, and particularly the red spots here represent the places where you have digestion of the peptide backbone. And so that's the real business end of the proteasome. If this is going around chewing up everything inside the cell, you'd have all kinds of problems. And so the regulatory parts of the proteasome are here, this cap structure at the top. What does that look like in a little bit more detail? Here we have our target protein in green. The little red pieces are the ubiquitin, which has been attached to this particular protein by the ubiquitin ligases we talked about last time. We'll talk about in just a second again. Once this polyubiquitin chain has been added, that's a target signal to get it to associate with the cap. Once inside the cap, you unfold this protein, put it into the garbage disposal. It's chewed up in individual amino acids. These can get recycled in terms of the rest of the cell. It's that process right here of unfolding, which also depends on having ATPases. And this is a now molecular structure. We have time today at the end to talk about how you get these molecular structures, of what that particular ring looks like. Here's that cap structure. It's really right at the bottom of this cap structure. That's what these individual proteins look like. They're ATPases, and they're basically serving as the opposite of those chaperones. Chaperones, you remember, were using ATPases to help fold proteins, now using ATPases to unfold proteins. That process will take this peptide, unfold it, and then pump it into the active site of the proteasome in terms of doing the whole degradation process. What is that label? Again, polypeptide, ubiquitin. It's an essential protein in those organisms that have ubiquitin. There are lots of bacteria that don't have ubiquitin. They have completely different degradation processes. But at least in eukaryotic cells, it's an absolutely essential protein. It's present in all eukaryotes, and the sequence is extremely similar in all of these different eukaryotes. And again, it's not terribly surprising. You can probably guess that from the name, ubiquitous. It's ubiquitous. It's all over the place, and present actually at quite high concentrations inside the cell. It's relatively small, only 76 amino acids, and that's really getting at the bottom end of when we talk about proteins. Usually when people think about proteins, 100 amino acids and larger. This one's smaller than that. And in the case of particularly the signaling for degradation, you usually have multiple of these ubiquitins which are attached to the protein that is going to get degraded. It's a covalent attachment that takes place. That covalent attachment happens through these lysine residues that are present on the outside of ubiquitin. What's happening there? We have a target protein down here with a lysine side chain that then hooks up to the lysine side chain on the ubiquitin and what's called an isopeptide bond. The reason it's called an isopeptide bond is because it's not in the regular chain of amino acids here. You have down here in your target protein, this would be your N-terminus and your C-terminus of this dipeptide. Of course, it's a much larger protein. And this peptide bond happens through lysine side chains. So it's that lysine side chain where you form this isopeptide bond. You can have either one ubiquitin which gets added, that'll be a monomeric ubiquitin modification, or most often you have chains of ubiquitin which get added. And those chains happen to be because you have ubiquitin which links to ubiquitin, which links to ubiquitin, etc. As I showed you before in that last slide, there are actually multiple different places on the ubiquitin molecule where you have side chain lysines that can be attached to through these isopeptide bonds to make these different branching points. And so here are those just highlighted in the primary sequence up the top, and then the tertiary structure down here at the bottom. Two of these linkages you can see here. If you have isopeptide bond formation at position 48 here on the left, or K63 on the right, you see this gives you a very different structure that you have of these ubiquitins which have been attached to the protein. And it's that structure which leads to the signaling where you actually go inside the cell. You go to the proteasome, or it turns out ubiquitin is used for lots of other things we'll talk about in just a second. We talked about this before, I just wanted to repeat it. How do you get this ubiquitin modification? You don't want to be modifying all of your proteins and all their lysines. How does that happen? Question, sorry, I missed you before.
So I'm wondering about the isopeptide bond.
Yeah.
Is that lysine bonded to another side chain, like nucleic acetate or something, or does it somehow connect up with the main backbone?
Yeah, so where does it hook up? So that isopeptide bond, when it forms, usually, again, there's some exceptions to this, but almost always is hooking up to the N-terminus of the ubiquitin, and then that N-terminus of the ubiquitin then can hook up to the next lysine bond, etc. But again, there are exceptions to that. So again, how is this happening? We've got three enzymes, E1, E2, and E3. Sorry about the nomenclature. It certainly wasn't mine to come up with in the first place. E1 is the ubiquitin activating enzyme. So it takes ubiquitin, as true in many, many cases, and we'll keep seeing this again and again. It activates it by putting a high energy bond onto that ubiquitin and linking it to this first enzyme. Then you have the E2 enzyme, which basically grabs the ubiquitin from this one and moves it over to the E3 enzyme. And the E3 enzyme is the really critical aspect of this whole process because this is where you get your specificity. It's how this E2 enzyme knows what substrate to modify. And so this is this, again, there you have your question about where it's getting modified in terms of ubiquitin. That depends on where your E1 is actually interacting with the ubiquitin. I mean, it's usually near the N-terminus, but not always. Then we get this stretch of ubiquitins. Again, this is depending on the E3 enzyme, where you're going to get either branched ubiquitin side chains or one linear ubiquitin side chain. This particular case, we've got all of them linked up together. This is really typical for proteasome-mediated degradation. But as I mentioned before, proteome-mediated degradation is by no means the only thing that happens when you have ubiquitin modification. So here, the second one right here, which is lysine 48, lysine 76, that's the residue that you're linking to in the ubiquitin. This almost always leads to proteasomal degradation, but there's a different lysine that you have. The top here is lysine 63. This is, again, a different lysine in your ubiquitin where you're making the side chains. This leads to signaling. Down here, lysine 33. It's important for blood cells, particularly immune system cells. Down at the bottom, lysine 11. This cross-linking is usually involved in cell division processes. So lots of different things that happen with ubiquitin modification. And we'll see this as we move on through this course, and probably even more critically in the cell biology course that hopefully many of you will be taking in the near future. So that's it for ubiquitin, protein modification, etc. Any questions before we move on to more general techniques and the current bane of my existence? Okay, so let's talk about centrifugation, because the centrifuge broke last time. That's my problem. So there are basically two different things that you can use centrifugation for purification for. Centrifugation is a really critical part of any protein purification process. And again, what this is about is separating different proteins from each other in order to be able to analyze them. So you can separate either based on mass or density. So mass per area. Pretty standard so-called differential centrifugation. You have your sample, you put it in a centrifuge, nothing else other than your sample, you spin it for a while, the heavy stuff goes down to the bottom, the light stuff stays up in the supernatant. And this will give you differential purification. We'll look at some examples of that in just a second. The problem that I'm currently having is this different process, here called rate zonal centrifugation in your textbook, usually just called gradient centrifugation, where you have a density gradient, and basically what you're trying to do is find where your particular protein is relative to the density of some other gradient. In this case, it's sucrose gradient. You now spin this particular tube. Again, the centrifugal force brings you to separation of these molecules based on their density rather than their particular mass. Once you have stopped this centrifugation, then really critically is you now take out different parts of what was present in this centrifuge tube. This fractionation process down here at the bottom. So this bottom piece, you have the one tube, the next part of that centrifuge tube, you have the next tube, and so on and so forth up to the top of this particular centrifuge tube. So this fractionation process that you get here, we'll see is critical for chromatography and all of the other techniques we'll talk about here. We talk about differential centrifugation. It's really easy. You have two fractions. You've got the pellet still in the tube and what's not in the pellet, which is your supernatant. So let's look at some of those pelleting processes. Here, start out usually by breaking open your cell. And when I say breaking open your cell, I usually mean the whole collection of cells. One case I actually worked with the horse pancreas. That was our first step that we did for just grinding everything out. Then you have this whole mixture. You need to separate that mixture. You do a relatively low-speed centrifugation. That's 1,000 times gravity for about 10 minutes. All the cells, nuclei inside a skeleton are down here at the bottom. Usually this is stuff you throw away. And then the supernatant has all of the proteins and interesting things that you're looking for. Then you can do a slightly higher speed, longer centrifugation. A lot of your eukaryotic organelles end up down at the bottom of the tube. Again, if you're looking for proteins, usually it's still going to be in this soluble or at least not pelleted part. You do higher speed centrifugations, 80,000 times G for about an hour. You can do this at most standard lab centrifuges. Here you've got almost all of the non-free proteins now in your pellet. All the proteins are now in your supernatant. Then, if you have an ultracentrifuge that works, you can spin at much higher relative G-forces for multiple hours, and now you start to get some of your macromolecular complexes that end up in the pellet, particularly the viruses, and this is the kind of thing that I'm trying. So this is a pretty crude way of separating out your individual proteins, but it's really fast, it's really easy. Again, assuming that things actually work. You can get much higher resolution. When I say resolution, that's basically pulling that needle out of the haystack. How big a handful do you take each time? You take a really small handful, that's a high resolution. Both hands full of that hay is going to be a lot lower resolution. So you need to break it down. The highest resolution you can get is that one particular protein, if you're looking for that one particular protein. So gradient centrifugation can actually give you much higher resolution and can also give you much more information in terms of what that particular protein or protein complex actually looks like. There are two different ways to do this. The one that I just showed you before is also known as velocity sedimentation. Here you start with your sample at the top of the tube and then spin it into this gradient below the sample. This now is a relatively short period of time that you do this spin for. This depends on the size and shape of whatever sample you're looking at here. So if you've got a relatively large complex that takes up a lot of space, that's going to go relatively slowly through your gradient. Whereas if you have a small or dense analyte that you're looking at, that's going to be much further down. Once you've done this spin, and that's what we have here in the middle, you've stopped it. Now, basically all you do is you stick a needle in the bottom of this tube and you collect the drops that come off of that. So, starting out here, the first drops you get will be here, next drops you get will be here, next drops you get will be here. Again, this is going to correspond to this fast sedimenting compound, which is what? Size-wise? Smaller. It's gone faster through the gradient. And then continue with your different fractions until you get up here to your larger ones. This is great, actually gives you a lot of information, again, about the shape and size of your structure. All the S's that we already talked about in terms of proteasome. 20S proteasome, 26S proteasome. That S is the sedimentation coefficient. Tells you about how big any of those processes are. We talked about the ribosome. We'll talk about 50S ribosome. We'll talk about 30S ribosome. That has to do with this sedimentation that's going on. The other kind of gradient centrifugation, and the kind I was trying to do overnight last night, is separating based on density. So it's not based on the size and shape. It's based on the density. And that process is basically spin as long as you possibly can in such a way that you end up reaching equilibrium. And so the molecule or complex that you're trying to, or virus that you're trying to separate, will migrate through the gradient until it gets to a point where its density is the same as the gradient at that point. And then it will stop migrating. And this will be true for all of the different complexes that you're looking at. They'll get to the individual density of the gradient that you're looking at least in theory, if the centrifuge doesn't stop halfway through. And so in this process, you end up with high density towards the bottom because your gradient has higher density towards the bottom, and lower density towards the top. So these are, and again, you often will take fractions, go off the bottom here, and hopefully by Friday's lecture I'll have a picture I can show you of a nice one of these density gradients. We shall see. I'm hoping to have that one today. But not. So, centrifugation, one way, and usually, very often anyway, the first step that you do in terms of protein purification, mostly to get rid of all the junk that you don't want. So it's a differential centrifugation that you use as the very first step. Sometimes you'll use one of these other gradient techniques, but that's much more often if you're going to do a final purification, which is what we're trying to do in this density gradient case, or really for an analytical purpose. What is the density of what I'm looking for? What is the relative sedimentation coefficient telling you something about the size? This is usually the second step after you've done your differential centrifugation, is now to try and separate the proteins based on some aspect of that particular protein that makes it different from the other proteins. So there are sort of four major processes that people use to do this. Either ion exchange, which is based on electrostatic interactions, and this just has to do with the surface charge of your protein. Think about the tertiary structure of your protein. It's going to have some charged residues. Those residues can be charged differently at different pHs. Each of the proteins can be slightly different relative to each other. You can use anion exchange for negatively charged proteins and cation exchange for positively charged proteins. We'll look at an example of that in just a second. Particularly for very small proteins and peptides, you can also use how hydrophobic the protein is. This is necessary with your hydrophilicity of the protein. How hydrophobic your protein, use so-called reverse phase, use organic solvents to actually separate the proteins relative to each other. The size of your different proteins you can use for separation purposes, gel filtration or size exclusion. And I would say 90% of protein purification that happens these days in most research labs uses affinity purification, and that affinity purification has to do with ligand binding. So a very specific ligand binding to the protein that you're interested in. And this is, of course, an extremely powerful technique because most ligands are usually extremely specific, and we'll talk about the case of antibodies. Of course, they're defined by their specificity. So let's look at a general case of column chromatography. I'm going to bring a column, but you get to it, we'll bring it next time. Basically, you have your solution, very often it's going to be supernatant from your differential centrifugation. You load it onto the column, either through gravity or using a pump. Once it gets onto the column, then it interacts with the matrix in some way, and then comes off the bottom, and some of these are going to come off the bottom faster than others. They'll depend upon the conditions that you use. And then, just like you do for centrifuge, you collect fractions and separate each of these individual fractions. So in this case, say this is your first fraction, the blue stuff is going to come out here, and you'll have some of your red stuff coming off out here. Now, that's great and wonderful. We can tell here that it's blue and red. Most proteins aren't colored. And so you also have to have a way of figuring out which of these otherwise colorless cubes has the protein that you're interested in. And so that's where you really have to have some way of detecting that look at all of these individuals as we go through. What kind of matrices do you have, or matrices? Here we've got gel filtration chromatography, or I like to call it again size exclusion chromatography. And basically, all that this is beads with small holes in them. The holes, small proteins going to get into, they get slowed down. So interact with this bead, that bead, that bead, that bead, that bead, etc. The large proteins have a harder time interacting with each of these beads, so they come through the column a little bit faster. Down here is your ion exchange chromatography, where you have mixtures of differently charged proteins. In this case, negatively charged and positively charged. The positively charged proteins are not going to interact with this column at all because your column matrix is positively charged. All the negatively charged proteins will. So you have positively charged proteins that come straight through the column without binding. Your negatively charged proteins now are bound to the column, but they're all going to have slightly different charges relative to each other. And so now you just increase an external ion, and so that's the ion you're exchanging with your protein for, and increasing usually salt concentrations. Now you can separate each of these different charged proteins, they're shown as cubes and spheres, relative to each other, relative to that concentration of that counter ion, the ion that you use in the exchange. The last step up here, you have to write the so-called affinity chromatography. This particular case has an antibody which is attached to this matrix, the beads that you have in your column. Here, your antibody should interact very specifically with your antigen, in this case, the red dot. This will now stick to the column, everything else does not. You can elute this by a number of different ways. In this case, going to a very low pH, and that low pH actually is going to denature the antibody, particularly the binding site on the antibody, those variable regions that we talked about. We talked about affinity and ligand binding. You can also use very high salt concentrations. You can put in something else that will bind to the antibody. Lots of different ways that you can actually get this to pull off. But this is a wonderful technique for going from usually a very complicated mixture with small amounts of what you're looking for to very pure amounts of that protein in basically one step, relatively quickly. Look at these in a little bit more detail. It's a different version, different textbook. Here you've got your positively charged beads with negatively charged proteins associated with them. Raise the salt concentration. Those proteins that don't have a very high amount of charge are going to be displaced. Higher salt concentration. Those proteins with a higher charge will be displaced. Gel filtrations, more what those gel filtration matrices actually look like. They're really porous beads where your small proteins can get into them and the large proteins can't. So if you look at elutions, these are just fractions here across the bottom. People talk about milliliters. Your large proteins will come out of the column early because they won't have associated with those beads. The small ones are going to come out much later because they have been interacting with the matrix. So, classic liquid chromatography. Again, this is just a very tip of the iceberg kind of analysis of looking at these things. Once you've done these processes, again, you need to assay what you've gotten off. What's in each of those colorless tubes that are down at the bottom of your fractions that you've pulled off. One way of doing analysis for this is very commonly used gel electrophoresis. How many of you have done gel electrophoresis? Many of you? Good. With proteins? Some of you? Good. But basically the process here is actually very similar to your size exclusion chromatography, only it's a much higher resolution process, and you can unfortunately use a lot less of your proteins. So it's great for analytics, it's actually not very good for purification processes. So the idea here is put your sample on the gel, put it in an electric field, separates based on interactions with whatever you have in that process. If you're talking about proteins, there's a big problem that you have with separation through gels, particularly if you want to get relatively high resolution, and that is the reason that ion exchange works so well. Proteins have heterogeneous charges. Usually what you want to do in an electrophoresis is you want to separate based on something other than a combination of charge and size. So the way that that's done, most protein gels, is by treating with detergents, particularly something like sodium dodecyl sulfate. Sodium dodecyl sulfate is wonderful because it's got this really hydrophobic part of the molecule and a really hydrophilic charge part of the molecule. So what kind of molecule would you call this?
Detergent.
Not detergent, but we talked about hydrophobic, hydrophilic, and
Amphipathic.
amphipathic. Exactly. It's a really nice example of an amphipathic molecule. How do you get this hydrophobic part of the molecule into your protein? Usually you need to denature your proteins. You pull it apart. Classic way to do that is to boil it. Put it in high temperature where you're getting those proteins to come apart. Protein comes apart in the presence of this detergent, and you get binding the detergent, and then what you end up with is a pretty uniform charge density. And so what that means is the charge per mass is really quite uniform. And so in that way, now you can take this and put it onto a polyacrylamide gel and get separation mostly based on size. It's not completely right, but it's really pretty close. And so now, with the sodium dodecyl sulfate polyacrylamide gel electrophoresis, everybody calls this SDS page, but the SDS is that detergent, polyacrylamide is the gel you're using, this is gel electrophoresis. You have your proteins. If they happen to have disulfide bonds between them, you want to reduce those disulfide bonds. That's the DTT or beta-mercaptoethanol, which we have in the previous slide. Boil these in SDS containing mercaptoethanol. Now they have these uniformly charged density, I should say, molecules. Now you can separate them in an electric field, and you get relative sizes of each of these proteins. This is a figure from your textbook. This is a figure from a previous textbook. I think it explains it a little bit better. But again, if you don't do denaturation in this case, you're not going to get nice separation based on size. Another critical thing that you need to do here is, okay, yeah, sure, B is bigger than C, which is bigger than A, but how are these relative to an actual molecular mass? So you also have to have standards. You have to have proteins of known molecular masses that you put on the gel at exactly the same time, and unfortunately, those aren't shown on this particular figure either of them for that matter. So, SDS pH, really standard, lots of hands went up. These are things that people do all the time in labs, and in fact we do some of it in our cell biology lab here as well. It's great, but it's not that high resolution, and usually the gels we're using are about this size. So it's hard to separate thousands, if not tens of thousands, different proteins on something about that size, and just separating it based on one particular aspect of the protein. A much more powerful technique in terms of higher resolution is something called isoelectric focusing. Also a lot harder to do, much more expensive. But what you do here is, again, it's not unlike your ion exchange chromatography, but here you have a pH gradient rather than a salt gradient that you use. And this pH gradient is in a gel. So here you have a stable pH gradient. That's what the blue shading here is supposed to be describing. In that pH gradient, you put your proteins at one particular position, at a particular pH, depending on the pKa of the side chains. Your protein is either going to be positively charged or negatively charged. So here, pH 10, your protein is negatively charged. It's the same protein, by the way. And pH 4 is positively charged. This now, in an electric field, will migrate until it gets to a point where there's no net charge. No net charge, doesn't matter about the electric field, it's not going to move any. And so this focuses, which is what this term is about, your proteins at their so-called isoelectric point, so the point where they have no net charge. And this is again, a much higher resolution technique than sodium dodecyl sulfate polyacrylamide gel electrophoresis, but it just gives the information on the isoelectric point. So it's analytically really nice, works well for separation, but again, relatively low volumes, and uses preparatively, but they don't work terribly well then. So probably the most powerful way that you can use isoelectric focusing is in combination with SDS Page. So SDS Page, separating based on relative mass, isoelectric focusing based on relative charge, the isoelectric point. You put these two together in two different dimensions, you can get extremely high resolution. In fact, in some cases, people will take a bacterial cell, break it open, do isoelectric focusing, do SDS Page, and then identify all of the proteins that end up on that particular gel. So this is an example right here on the right-hand side of a typical, actually really, really nice textbook, isoelectric focusing SDS Page two-dimensional gel electrophoresis, also known as 2D gels. A lot of people just talk about these as 2D gels. So it's isoelectric focusing followed by SDS Page. Here, you've done your isoelectric focusing, and then all you do is take your isoelectric focusing gel, it's usually done in a tube, lay it on one side of a polyacrylamide gel, and separate it based on SDS Page. So theoretically, really straightforward. In practice, it's really an art to do these things. And these gels are often about this kind of size. They're actually relatively large. But each of these individual spots now on the gel represents one protein. Now, multiple spots can, however, represent the same protein if, for instance, a protein has a different charge on it. Now, what's one way you get a different charge on a protein?
Proportion of
So change the pH, yeah, you'd have a different charge, but we've already had that separation here. So this is now the isoelectric point. Last time we talked about kinases. What do kinases do? Phosphates. What do phosphates do? They've got this really powerful charge on the outside of the protein. That can be the same. So what is it usually phosphorylate? Phosphorylating what? 3 different amino acid side chains. I will expect you to remember this.
Lysine?
Serine, threonine, and
Serine.
tyrosine. Those have charge? No. So you phosphorylate, you change the charge. So 2 spots, which should have very similar molecular masses to each other, say like these 2 down here, about 20,000 molecular mass. These 2 could be exactly the same protein, but one's phosphorylated and one's not. So that's one thing. So each spot is going to represent a protein, but some proteins can be present in multiple different spots. Particularly we see the smear, something like this, very often the same protein which is going to slightly modify. Each of those spots is great, it's wonderful, and each is an individual protein, but how do you know what protein it is? So this gets to that assay process that we have at the end. You have a band on a gel. Now, we didn't talk about how you get those bands on the gel, how you get spots on that previous two-dimensional gel, but these are usually through very non-specific staining techniques. So some of the original techniques, the original stains that people used, are actually things people used to stain clothes with. Pretty standard dye processes. It turns out they bind proteins really well, and what are most, at least, before we get all these synthetics, but cotton, lots of proteins, lots of the other dyes that would work well with some of these textiles work really well for protein binding each other. So if you want to identify the one particular protein that you're interested in, you happen to have an antibody for it, you don't always have, but if you do, a really powerful way of detecting that one particular is through Western or immunoblotting. And all that this is you do your regular STS page, you transfer that STS page and the pattern of bands that you had on there to a solid surface. And the reason for doing that is twofold. One is that you concentrate, because you've gone from a gel which has a thickness, which is surface. And the second reason is that now it's accessible to whatever you want to put on it. In this case, it would be an antibody. So you do this transfer, then you bind your specific antibody to this antigen, the ligand for that particular antibody, the one that you're looking for, and then you still need to detect it because the antibody is still colorless, it's tiny. And so almost always you'll use what's called a secondary antibody. That's an antibody that interacts with the first antibody. So ligands, antibodies can bind to various different proteins, enzymes, etc. But of course antibodies are proteins. So you can also get antibodies that will bind to antibodies. And particularly these usually bind to the heavy chain part, the stable, doesn't change very much part. And this is almost always attached to some way of detecting that particular second antibody. There are lots of different ways. This case is used chromogenic detection, which is where you put an otherwise colorless substrate onto your Western blot. This antibody is conjugated to the enzyme. That enzyme will react with the substrate, give you reactants. Those reactants will then have a color. Or in many cases, it will also be a light-driven reaction. So it gives you light. That light can be detected either by an imaging system or, for us ancient people, also with film. Another way to do this is probably I think the least intrusive way of doing it is using radioactivity, which also you can use photographic film to detect. There are way more radioactive isotopes than are listed in this particular table, but these are the ones that are most commonly used in biology and molecular biology, particularly our friend P32, phosphorus 32, and sulfur 35. And the reason for that is that phosphorus is present in all of our nucleic acids. You think about all the kinases, they can put this phosphorus onto lots of different proteins. And radionucleotides are really nice because they don't change what you're looking at very much. So very, very small changes. Enzymes usually don't seem to care if you have a radionucleotide as opposed to a non-radionuclide which you're using for your particular process. So for instance, if you're using DNA, you have radioactively labeled ATP and non-radioactively labeled ATP in your same reaction, the DNA polymerase doesn't care. They can use either of them. And the same thing is true for pretty much all enzymes. So these are really nice ways of incorporating a nice label into whatever you're looking for. Of course, you still have to deal with the radioactivity, but I actually prefer dealing with radioactivity than a lot of other things. And we can talk about offline why I think radioactivity is the way to go for a lot of biological experiments. One thing that you can look at here is what's called the pulse chase experiment. Here, we're looking at particular proteins, which has been labeled, remember correctly in this case, yeah, with this a carbohydrate addition, so you have a radioactively labeled sugar, which gets added to your particular molecule. Here, precursor protein gets labeled by this particular radioactive precursor, labeled for a short period of time, and then you put in large amounts of the non-labeled precursor that went into this whole process. So in a normal case, which is up at the top, you have rapid incorporation of this radio label, and then over time, this will exchange with the non-labeled precursor that you put into the reactor. So as I say, the pulse is a short amount of incubation of your radioactive substrate. Over time, that decays. And you can actually get really nice kinetics in terms of how you get this degradation taking place. On the other hand, this is the experiment shown down here at the bottom. If you have a mutant protein, it actually doesn't get converted to your mature protein here, and you can actually see this with your pulse chase experiment. This guy is much more stable, and usually your mutant protein here, in this case, will say be one of the side chains that you think your carbohydrate is getting linked to. So, really nice to be able to do this. The way you detect the radioactivity is, again, either with imaging systems or very often just photographic film, which you put down on top of, in this case, it's an STS page gel. So in that STS page gel, STS page is denaturing your protein, but that covalent linkage that you have to your radioactive and then later non-radioactive polysaccharide is still going to be there. So it's a nice way of doing these kinds of experiments. So in the last 10 minutes, we'll talk a little bit about mass spectrometry. As I mentioned at the very beginning of the lecture today, has really revolutionized the analysis of proteins, particularly protein mixtures and all of the proteins in one cell or one particular organ, etc. Whole idea of mass spectrometry is you need to ionize whatever you're looking at. Once you have these ions, then you can separate these ions relative to their charge to mass ratio. This happens in very, very small amounts of your analyte, and it's generally a non-quantitative method, really a qualitative method, which is, you can basically tell it by spectrometry. You're literally measuring the mass to charge ratio of the ions that you make. So there are two important aspects about all mass spectrometry. One is the ionization technique. In this case, it's a laser which will ionize the analyte that you're looking at. So this is the matrix associated. So matrix, which we have here, is the metal target. Laser dissociation ionization. Basically, you blast a laser at your sample here that will ionize a very small part, actually, of the sample that you're looking at. And that then you can accelerate in your mass spectrometer. Then you have a detector. This particular detector is actually usually a very short distance away. We're talking millimeters to sometimes fractions of millimeters. But this distance that it goes here, under, again, a very strong electric field, these guys are going to go faster, the small ones, larger ones, and it turns out that you can get very, very, very high resolution here, to the fractions of mass units on this particular kind of mass spectrometry. This is great for doing very specific masses and usually relatively small analytes. So each of these peaks here, this is going to represent a particular point where you have detection of this analyte, etc. Another way that people are doing mass spectrometry, and this is probably the much more common mechanism, and also the one that's used in proteomics, we'll talk about right at the end here. This is the so-called electrospray mass spectrometry, where you have a, again, usually a very small amount of liquid, which you pass through very high voltage narrow needle. This will give you an aerosol to start with highly charged droplets. Those droplets evaporate. You end up with these ions. Again, an electric field. These are accelerated into a mass analyzer. This mass analyzer is really great because you can actually say, I want a particular mass, so I'm going to look for such a mass, which will be a mass over charge analyzer. I want to analyze and sometimes even separate a particular mass. You can do that with these particular mass spectrometers. And then there's a detector, again at the end, which is very similar, gives you a mass to charge ratio. This is one process that you can use this for. Here we have a protein sample, separated it through STS page. Very often you literally will cut out the band from your STS page, take this. Usually the next step is to break that particular protein down into smaller pieces. because if you have smaller pieces, you have an absolute resolution, a certain number of mass units that you have on your mass spectrometer. You've got a smaller piece, you're going to get a much more accurate determination of what that piece is. In your mass spectrum, you end up with a spectrum here, which you then can convert to what your M over Z is. And then the really critical part of mass spectrometry is not so much all of this aspect, making your protein, separating the base on mass. It's really the data analysis step at the bottom here. So it's the advent of really fast, powerful computers that really allowed this to take place. Go back, sorry about that. Back a slide. This is what that data looks like. And so here, relative numbers of ions, but again, it's just a small proportion of what you're looking at, which is actually there. Here we have one particular ion, mass over charge ratio of 836.47. You can actually now take this, again, because of your mass analyzer, take this one particular ion and then split it into multiple different ions. What's called the MS, so mass spectrum of a mass spectrum. Take this one, split it into lots of different ions, and those ions are usually made by breaking this first ion in each of the individual peptide bonds. And each of those individual peptide bonds is going to give you a whole stretch of different masses, starting 261, 421, etc. And the difference between each of these actually corresponds exactly to an individual amino acid. And so you can use this process for sequencing your peptides. So let's talk about just that one example right here. Here's our protein at the top. We've separated out on our STS page. We now chop it up into little pieces. We use either a known enzyme like trypsin or a chemical like cyanogen bromide. Trypsin we talked about before. This cuts right next to long positively charged side chains, so next to lysines and arginines, so all the lysines and arginines here. Cyanogen bromide, which we haven't talked about, that cuts next to methionines. So here, this is going to cut next to particular methionine residue. And then you analyze all of the peptides you get made from that. You get exact masses from each of them. And so here, and also the sequence. So we've got a sequence of AGKPLM, IHE, BGR, etc. You take these peptides and then compare them to these peptides, see where you have overlaps between the two. And so here, AGK is an overlap between this peptide that you got from the trypsin and this peptide that you got from cyanogen bromide. You can walk your way through here and you end up with the whole sequence of this peptide that you started off with up here at the top. And so it's an extremely powerful way of getting the sequence of individual proteins and also identifying each of those individual proteins. And we'll talk about that protein identification process on Friday.